meeting, guys.
Yeah. So this is lesson 12. And I'm gonna need, honestly, like, I had a really, and it might just be because I didn't, like, thoroughly read through the notes, but, like, I looked on the PowerPoint, I did look through the notes, and, like, I tried to read through everything that I needed to. And I still, a lot of the questions, I don't understand why, like, the first one even, I thought I got that, and I looked on the key, and it's not right.
It's okay. I hardcore struggled. I don't know a lot of the answers.
You hardcore what?
Struggled.
Oh, struggled, got it.
Struggled. As in, like, ah!
Yeah. Okay, well, let's start. So,
Cool.
I can read number 1 if you guys want. It has this plasmid. So suppose that the PBR322 plasmid is cleaved with the restriction enzyme PSTL. Suppose a PSTL digested segment of mouse DNA is then ligated into the PSTL site in the PBR322. This recombinant DNA molecule is then used to transform E. coli cells. So first, like, seriously, I feel stupid. So if anybody has any, like, anything that would help me understand more, that would be great, and I hope that I'm not the only one. But, like, I tried to understand what exactly a recombinant gene was. So I know that in order to clone a gene, or in order to remove a gene that you want from a chromosome, so you remove the gene and you insert it into a vector molecule, so like a plasmid would be what we're looking at here. So that a recombinant gene is when you take a section of a gene from one chromosome and stick it in a vector like a plasmid, right? That's what it means.
Yeah.
Okay. So A says which antibiotic should be added to the medium to select for cells that have incorporated a recombinant plasmid. And I saw here that the plasmid is tetracycline resistant and ampicillin resistant. So in order to, so which antibiotic should be added to the medium to select for cells that have been incorporated? So to see if, I don't know, I thought you should add both because it looks like this plasmid is resistant to both, but the answer key says that we should only add tetracycline, and I don't understand why you would choose tetracycline over ampicillin.
Hmm.
Does anybody have any idea? So, like, I know that we were looking at, um,
I kind of wish they had this figure like in the PowerPoint.
what?
Or, well, maybe they kind of do.
She said she wished this was in the PowerPoint, like more, like better explained.
That's the thing. I had a really hard time with these notes and PowerPoints because I felt like, yeah, it gives us an idea of what, but with these questions, it was just hard because maybe I just didn't read the right parts because I didn't go all the way through like I should have, but I don't feel like it really addressed some of these things. Like in depth to where I would understand, okay, this is what it's talking about.
I did not like this unit.
So we're looking at, so I'm going to go, anybody jump in or stop me, and if you guys I just want to try to understand this. But suppose the PBR2 plasmid is cleaved with the restriction enzyme PSTL. So that means that does that mean that the PSTL was inserted into the PBR322?
PBR 322 plasmid is cleaved.
That's what I. And then which antibiotic should be added to the medium to select for cells that have incorporated a recombinant plasmid? I would think, because that's what I'm confused. So the recombinant plasmid, so it says which antibiotic should be added to the medium to select for cells that have incorporated a recombinant plasmid. So the recombinant plasmid is the PBR322 plus the added DNA from the PSTL, is what I understand. So we know that the PSTL segment is ampicillin resistant. So I would think in order to test or select for cells that have been added into the recombinant plasmid, so the PSTL, you would apply ampicillin, because then it would show, I don't know.
No, I can get what you're saying, because like why would you use tetracycline if you're trying to select for that segment?
Yeah, that's what I don't get. Like, I feel like I'm going in circles here, but I'm hoping like if I talk through it, then something will click, but it's not clicking.
Self-digested, the segment of the mouse genomic DNA is ligated into.
Let's see here.
Because, wait, which antibiotic can be added to these?
Okay, so markers. So what it's talking about is the selectable markers. So it says plasmids contain additional genes that confer an antibiotic-resistant phenotype on the host cell. So I think that would be the BAMHI tetracycline-resistant gene. Thus, by growing host cells in the presence of an antibiotic, you can ensure that each host cell contains a vector or beta globin gene. Don't really understand that, but okay. A common selectable marker is ANPR, which confers resistance to the antibiotic ampicillin. That makes sense. Thus, a cell that is resistant to ampicillin contains the vector. A cell that is sensitive to ampicillin does not house the vector. So we're looking at PSTL, so it's ampicillin resistant, so we know that it has the AMP-R marker. So why would we test with tetra?
I'm highly tempted to just email and ask, because I'm really useless when it comes to like all this, so I feel bad that I can't answer anything.
No, it's not your. I'm just trying to like get kind of discussion going, because if you guys have any other thoughts, if not, I mean that's what we might as well do, because I've looked in the notes.
Eileen!
Hi.
Maybe you can help.
They missed me. No. Just kidding.
Where are you?
I'm at home, like home.
That's cool.
Yeah. And my friend Garrett is here, and he's laughing because I'm just like talking to my computer, and it looks really weird. And then he hears your voices, but you know.
We got stuck on the first problem.
On the first one,
Yeah.
Yeah, we
Because I
got
keep
stuck
copying
here.
Horrible.
I
hold
don't
on.
think so.
Let me pull up all my stuff because I did okay on the second and the third one, like the ones we turned in. Like I was
Yeah.
fine.
See, I'm just in trouble.
Let's see. Did you guys have trouble with those ones too, though? The ones that were due?
I didn't have too much trouble with them. I don't really remember them right now. I gotta pull up.
Number 3, I was like totally fine. Number 2, I'm hoping I did alright. And then number 1, I'm like,
Yeah. Hold on. Garrett, do you know about genetics? Oh my gosh, shut up. So conceited.
What?
Just kidding.
I would say narcissistic.
Okay, so narcissistic. Whatever. Okay, let me look you guys. Oh, I don't know about this stuff.
Phenomes.
yay, we're all useless then.
Okay, yeah, I don't know about this stuff either, you guys.
Okay.
Oh, we're all screwed. That's okay. Oh man.
What is it?
Would you answer an email?
Is this a PCR? Is this what this is? The first one?
It's a vector. It's just talking about how. So we're looking at what we're confused about is we're looking at the PSTL segment that was added to the plasmid. And A is asking which antibiotic should be added to the medium to select for cells that have incorporated a recombinant plasmid. So we thought, or I thought, I don't know what it is, but,
Sorry, this is so funny.
This is awesome.
we thought that like
My name
since it says on the sheet PSTL is ampicillin resistant, that it would be ampicillin. But it's tetracycline.
Wait. Hold on. I don't remember this one at all. I just focused on 2 and 3. Let's be real here.
I mean, we could just send her an email.
Where does it say that it's, oh, it does say that it's ampicillin resistant. What antibiotic should be added to the medium to select for cells that have incorporated a recombinant plasmid? Well, okay, hold on. Oh my gosh. Is it? It's on the couch. If it's, I'm really confused about this too. Hold on. This is like it,
See, I can't even find tetracycline on the notes.
Well, it says it's not in the notes, but on the plasmid, the BAMHI and XMALL segment are tetracycline resistant. So I don't know why you would test that, because then it seems like you're testing more to see the characteristics of that segment versus the segment that you added to the plasmid in the first place. We can just send her an email.
wait, so what's the medium
Uh,
for this? Does it say what the medium is that we're using? Or the media? I don't know.
not really, no.
Okay. Hold on though. So if it's, which antibiotic should be added to the medium to select for cells that have incorporated a recombinant plasmid? Hold on, let me just like look at the notes again, because I don't know why, but I just like the notes about it. Hi, bird. He's not happy. He's trying to sleep. Oh, Caesar.
Man.
Um,
The main thing that I saw in the notes that I read to them talked about, it's on the page. That talks about vectors and plasmids. And it says plasmids contain additional genes that confer an antibiotic-resistant phenotype on the host cell. Thus, by growing host cells in the presence of an antibiotic, you can ensure that each host cell contains a vector or a beta-globin gene. A common selectable marker is AMPR, which confers resistance to the antibiotic ampicillin. Thus, a cell that is resistant to ampicillin contains the vector. A cell that is sensitive to ampicillin does not house the vector. That's all I got. That's all it really says.
this is so confusing.
I think we should just send her an email, because if not, we're just gonna like go in circles.
That's true. Can I just like read through the rest of these though? Hold on. So how do you explain the presence of bacterial colonies that are resistant to both antibiotics? Oh my gosh. I just need to reread the notes.
I don't feel like I don't know.
Or
I
rewatch the video. Was there a video on this?
No,
Oh my gosh.
or maybe I didn't really watch all the videos, so I can't really say that. But I read through the titles of the video, and it didn't look like there was any.
That's true. It's really true.
No, it gets to the end of the semester. There's no time. I don't know. Whatever. Yeah, that's fine. We can email her about it and figure it out later because I have no idea.
So
Just for time's sake, do you want me to email like now?
that would be
Sure.
awesome.
Yeah, go for
Okay,
it.
I will do it. And then when she emails back, I'll email it to you guys.
Okay.
So B says, how can you explain the presence of a bacterial colony that are resistant to both antibiotics? And the answer is, I don't remember what I wrote, but it wasn't right, whatever it was. But the answer on the key is bacterial colonies that are resistant to both ampicillin and tetracycline do not contain mouse genomic DNA inserts ligated into PBR 322 plasmid. I don't understand what. Was there a section on mice that I missed?
I don't know because none of this sounds familiar to me either.
Wait, I spaced
we
out.
just, like,
Where are we?
Still on number 1B.
Oh,
I vote we just like skip the whole thing because I honestly none of this makes sense to me. But
Mouse genomic DNA inserts. Okay. That's not what I wrote.
really good. Oh, freak, it's nothing.
Does anybody else have any comments on D?
no.
I'm just as confused as you guys are.
Okay. Well, I'm glad I'm not the only one, because I was really like getting aggravated because I was looking through and trying to figure out why I wasn't understanding. But I mean, after this, I can't really like. I was gonna get the second portion of questions done tonight, so I was gonna read through everything again. So maybe it'll make a connection somehow. But C says you decided to adjust your recombinant DNA molecule, the vector plus the insert, with the BAMHI and echo RI, and then analyze the resulting fragments on an agarose gel. How many bands will you see on the gel?
The key says 2, but I don't know why.
So
I
I
wonder
would
it's that
add
Sanger method of DNA sequencing.
Oh, is that what it used?
I'm guessing because it looks like it
Oh,
sequenced
because it
it
has
from
the
gel
bands.
and bands
Bands and
marking.
make her dance. Bands and make her dance. Okay, good night, Garrett. Oh man. That song's super
Why
stuck.
two? I don't know.
Huh?
Said why two? I don't know.
I don't either. So BMHI, is that even okay? So studies are blah 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 with BMHI and Echo RI. So I would think that would take one band out, two bands out, and then that would leave. I thought it would be 4, but what do I know? Or maybe no, I don't know. I have no idea. This is aggravating. And gonna go on YouTube. I wish I could make it private somehow. Like, because when somebody like looks up genetics, okay.
And this is what they get, a bunch of girls who don't know what they're doing.
Okay, so D, you decide to digest your recombinant DNA molecule with BMHI and PSTL, and then analyze the resulting fragments on an agarose gel. How many bands will you see? It says 3.
Still don't know why.
So whenever it says digest, so whenever you digest with a certain
Wait, is this gel electrophoresis? Is that what it's supposed to be?
Yeah,
Oh,
it's an electrophoresis gel. That's why it keeps asking for the bands.
okay. Oops.
Okay, so the digest does it, and this is in the notes, but for some reason it just the digest targets. So I felt like the digest. So if it's BAMH, for example, BAMHI, then that would be the one that it would kind of like it would target it to cleave, right? To take out. Is that right?
I don't know. I just want to skip number 1 altogether, because I have no idea.
Who wants to take 2?
I'll do it. I have no idea anything about number 1. I'm so confused about that. I just need to go back through and read everything and figure it out, but an email wouldn't hurt. But anyways, okay, let's do number 2. So this question concerns PCR and DNA sequencing. Describe what would happen in a PCR experiment if you only used 1 primer instead of 2 primers.
Where's the answers?
They didn't show up on here, but basically, whatever, we can just talk about what we have, because I don't know why, but there's no answers for number 2. They're on, like, all the other ones, but yeah. So for this one, I just put, like, if you only had 1 primer, wait, actually, let me read what my actual answer was, because it's going to make a lot more sense than if I try to explain this. Okay, I put, if you only use 1 primer during PCR, only 1 part of the template strand would bind to the replication strand, and it would not form a complementary strand, because the 2 strands would still be separated, except for that 1 part with the single primer.
So like the template would still separate, but only the template strand, only the one template strand with the primer would replicate.
Yeah, but it like wouldn't, but it would, like those primers are like there to hold it in place, right?
Yeah.
and then like replicate, whichever way they're gonna replicate. But like if you only have one, it's gonna be like still unzipped pretty well. So it might like replicate a little bit, but I don't know. Like it's not gonna have two primers holding it in place
Yeah.
so that it can replicate completely. It's gonna be like unzipped pretty much. So you might get like a few like nucleotides replicated, but then after that, I don't think it's gonna replicate anymore.
Okay. So if that was the case, what did you get for B?
So I got that it was 32,768 strands.
Okay.
Wow.
I got a totally different number. I feel
How did you do it?
But did you do?
That's the same number I got.
Two, but I did molecules versus strands.
What?
What was your answer, Shannon?
I'm kind of embarrassed to say it, so I'm not really sure if I want to say it now.
Just say it, because I could be wrong.
655,360 copies.
Okay, what did we do? Like, how did you do yours?
Wow.
I got 16,384, but I think that's Eileen's number divided by 2. I think. I didn't do it, but I didn't do the math, but sounds like it.
I just read the notes and it said that it would just double the number each time. So I
Yeah.
just like, multiplied it by 2 all the way up until it had the power of 15, and that's why I got the 327 whatever thing.
I just did
Well,
the 15 to
same
the
as
power
mine.
of 2. Which
Can
got
you
me
give me that number
225.
I did 2 to the 15th power.
That makes sense because it's 2 to the n is the rule, right?
I don't know, but that makes sense. If it's doubling every time, it's going to be 2. Yeah, it would be 2 to the n.
I thought I did 2 to the 15th, but I'm also really bad at math. So
I just put it in a calculator and put 2 to the 15th power. Like that's literally all I did.
What was the number?
37 something. Oh, 32,768. So we just put 2 to the 15th.
now I got that. Okay, yeah.
Okay. Got it.
That makes more
I
sense.
got it.
Okay.
I mean, I still could be wrong, but that's what I used. That's the math that I did.
No, the whole time I was thinking, like, there's a rule for this, but whatever. But with it being only one strand, though, because the question says...
It
like-
still doesn't matter because so you always start with one strand, like the examples in the PowerPoint start with one strand
Yeah, okay.
and the notes and stuff. So that's how I did it, was like I used the notes in the PowerPoint that started with one strand and it doubles every time. So yeah.
Okay.
I mean my math could be wrong, I guess, but like I feel like it still should be 2 to the 15th. Because it's after 15 cycles.
Yeah, I think that's right. It sounds right.
Okay.
Yeah, I tried. Yeah, I wanted to do 2 to 15th, but I don't know. So I think I just screwed up entirely. So yay.
That's okay. Like seriously, my math could still be wrong, but like that's just the way that I did it. So I mean, as long as we know how to do it, like I think that's what's more important than getting exactly the right number. I mean, the right number is still important, but I don't know. I always mess up my math, so whatever. Okay, so for number or whatever 2C, it says we usually think of enzymes being most active around 37 degrees Celsius. In PCR, however, DNA synthesis occurs at 72 degrees Celsius during each cycle. What's unique about DNA polymerase that's used in PCR? And I put that the DNA polymerase used in PCR is derived from a thermophilic bacterium whose DNA are not denatured at the usual 37 degrees Celsius.
Yep.
That's important for PCR because high temperatures are used that
Yeah, to split
normal
them.
polymerases wouldn't be able to stand.
Yeah. Okay, and then D says dideoxynucleotides play an important role in DNA sequencing. What's the structural difference between a nucleotide a deoxynucleotide, and a dideoxynucleotide. And for that, I have nucleotides are like are standard, structurally speaking, like the nucleotides we learned about a few weeks ago when we were learning about like the structure of DNA. It's the same nucleotide. But then the DNTPs are missing the hydroxyl group on their 2' carbon, and DTNTPs are missing 2 hydroxyl groups. One is on the second, or the 2' and one is on the 3' carbon.
Oh.
Yep.
Missing 2 OH groups.
Yeah, I think she posted a little picture of it on one of the teacher's notes on the discussion boards.
Okay.
See, I thought, for some reason, I thought DNTP had, in my notes I have that DNTP has an OH group and DDNTP doesn't. And I'm thinking that sounds like RNA versus DNA. Like,
Yeah.
but
I
that,
think I thought that at first, and I had that down as my answer at first, and I don't know why, but I like thought that I read that in the notes, and then I went back and reread it, and I found the real answer. So
okay, I'll have to reread that. Sweet. Who wants to do 3? Because I don't get that one at all.
I guess I can do 3. Okay, basically listed below are cell restriction enzymes along with specific nucleotide sequences recognized by the restriction enzyme. I can guess through the table. Let's assume that we'd like to digest chromosome 1 from the fruit fly. And for each of the above enzymes, we assume that Drosophila genome is composed of 30% iodine, 30% T, 20% G, and 20% C. What is the average distance between each of these restriction enzyme recognition sequences? At first, I really didn't get it, but I had a glance at the key for it. Now it kind of makes more sense. Basically, every, so with A, the total amount is 278 base pairs. That should be the answer.
Where did we
A.
get those numbers from, though?
What you're doing is that for every, like in the first recognition sequence, the A little thing that says AGCT, that is 0.3 times 0.2, 0.2, 0.3.
Why? Where do we get those numbers from?
It just says like how much they are composed of. It's the percentages that they said.
Oh.
Yeah. And then you take that sum and then you divide 1 by it. And you just do the same thing over and over again.
Okay. I was getting so aggravated. I was like, where are we getting these numbers from? Like, they're asking for distance. I was looking for, like, a measurement of distance. Like, I don't know, micro somethings.
yeah,
Do you
it's
want
just
me
the
to read?
average.
Yeah.
Yeah.
Do you guys want me to read the totals for each of them?
Yeah, just go for it.
Yeah. Okay, so A was 278 base pairs. B is 625 base pairs. C is 3,086 base pairs. D is 6,944 base pairs. And E is 390,625 base pairs.
What was A, B, C, D?
D,
Uh-huh.
6944.
Okay. Right.
Yeah, and for these, when we calculate it, we just round it, right? Just double checking.
I think they gave you an exact number. Like you get your
Well, one of them gave me exact number, but then other ones like gave me like 277.78, for instance. So
really?
yeah.
Okay.
Okay, you round it by your closest decimal.
Okay,
They
just
didn't.
double checking.
Yeah.
But just to clarify, was this in the notes?
I
think
don't
so.
remember
Nope.
seeing this in the notes. I had to look at the key to figure it out.
I
I
did
was
too.
Me too.
But it makes sense.
Not
It's a
to
lot.
stupid people like me.
Well, like, I mean, yeah, like I still had to look at the notes, but I
I
don't
was
know.
very angry. Okay. Maybe I just don't have enough critical thinking skills to figure it out on my own.
It's not it.
If this question was on the test, I wouldn't mind it.
Yeah, it's
At least
easy
that I know,
now.
like, hey, I can do this.
Mhm.
This is what I can do.
Yeah.
Yeah. Okay, who
I
wants to
feel like okay.
Sorry. Go ahead.
No, I was just gonna say like, had I not known how to do this at all, and this was a test, I would have been like trying to find a gun and shoot myself in the head. But after looking at the key and then doing it, and then if someone were to give me a test, then I would have been like, okay, this is easy. But it's just that had it not been for the key, this would have been a very, very stupid question.
Okay.
That's all. I just wanted to rant.
Who wants to do 4? I think I get this one, maybe, kind
Because I don't.
I can read it, but I don't get it, so.
of. Well, I guessed, but I don't know what the key is. Go for it.
The key has a photo. Anyhow, yeah. So it says, you wish to use DNA sequencing or Sanger, whatever method, to determine the nucleotide sequence of the molecule shown below. And then there's the sequence. I'm not going to read it. The primer annealing site indicates the region of the template DNA strand where the primer will anneal, form hydrogen bonds during the sequence reaction in order to generate the newly synthesized strand. Note that the newly synthesized strand will be complementary to the sequence above. After DNA sequencing using the Sanger method described in class, the DNA fragments were separated by electrophoresis on a gel. What would the banding pattern look like on the gel?
Maybe I don't get this. Because what I did, the only thing that I could figure out how to do was, and this is probably totally wrong. I mean, it kind of looks like it's about the same. So, the DDGTP has the least like there's only 2 G's in that strand. There's 4 C's, 4 A's, 4 T's. This could just not be how you do it at all, but this is how I did it. And so I thought because there's more G's present or there was the least G's present, that it would be carried farther. And then I thought that the rest are all 4s. There's 4 C's, 4 A's, 4 T's. So I thought that they would all be present at about the same area.
But are you looking at the key?
Yeah, I'm looking at it. I know it's wrong. That was my thought process.
And I don't know about the distance thing, but like because it's a complementary strand that we're doing, so if there's only 2 G's, there's gonna be 2 DDCTPs because that's the complementary
Oh, okay.
thing. Like, yeah, that's like the complementary whatever those deoxynucleotides are.
So instead, we're gonna say there's 2 C's
And 4 G's, and then 4 A's and 4 T's. I don't know about where they're placed on that thing. I don't know about placement, but I know, like, you know, how far they travel and stuff and where they hit, but...
Well, I just have no idea why it looks like that, though. To me, like, you'd need more information. Like, the base pair thing on the upcoming questions makes a lot more sense to me.
I got it.
Yeah?
Okay, so just like what she said, like how like it has to be the opposite. So if you go from down to no that. So if you go from the top to the bottom, so if you go from GG, so that's why on the column where it says DDGTP, that's why there's the X there, because that's the opposite. And then the third one is supposed to be like, you know how it says on the strand CCG, but then you go GGC, and so you go GG on the DDGTP strand, and then you go
okay.
GDCTP on the third, and then you go, and then on the strand it says AA, and so instead of AA you go TT, and then you go on the other side and then TT, and you just go throughout the whole thing like a little jigsaw. Oh, I got it! I'm sorry, I got really excited.
Okay, that makes
So,
sense.
That makes
opposite.
more sense.
So it's going to be G.
Got it!
So up here, GG. And then C. Oh, it just goes in order.
A,
Yeah,
A.
you write out your complementary strand and then you mark them on there in order.
Oh,
Oh,
sweet. So that is with what medium? Because we're going to be asked.
That's the gel electrophoresis. Oh. I don't know. What medium?
Like what? Because you know how she asks for different things. Like there's the circular plasmid restriction thing. There's just plain linear restriction. We're looking at
I don't know.
electrophoresis. That's electrophoresis. So then it just goes in order for that. So we need to remember that.
Yeah.
Okay. Sweet. Thanks, girl. Who wants to do 5? I kind of get this one, I think. I haven't looked at the key, but
The key doesn't.
have anything.
Yeah. It just
So-
says, like, draw the linear restriction map, draw the circular plasmid restriction map, which that's one I don't know how to do.
Yeah, that one's hard. I found a YouTube video, and it kind of makes sense, but, like, not easy. And I don't know if I got it right. But for the other ones, for, like, A, you just have, like, it tells you, basically, it tells you what the control is. It says, suppose that your control is at 100, 500, 1,000, 2,000, 3,000, and 4,000. So what I did is I drew like a rectangle, and then I made 4 wells, so that includes the control and each like BMHI, whatever the rest are. And then I went from the highest because the negative electrode is up by the wells and the positive is down at the end of the plate. So you start, like, I wrote out all the things like 4,000, 2,400, 1,600, 1,200, 800, 400, 100. And then so you mark a line where each of the control bands were, and then you mark a line where each of the BAMHI bands are, and then you mark. So it's like it tells you where to mark, is what I understood anyways.
Okay.
And that's the same pretty much for B.
So you're just, like, graphing it, basically?
Yeah, that's what I understood.
Okay.
It's just C that I don't understand at all. Like, I know that you start by, so you draw a circle and then you choose one of the genes to start with, and you put it at the 12 o'clock mark. So, like, they all should, each of the segment numbers, so like the base pair numbers should be represented in the circle. So we know that like the full circle is what, 5,200 base pairs, and then we know that. So each of, I wish I could send a YouTube video because it. Let me see if I can find it and like show it to you guys so you can look it up. Because I'm all just confused, y'all. So give me 2 seconds.
If you want, you can send the YouTube video on the discussion board thing. So like after you post a link, you can post like the thing, like the little video that you were talking about.
Okay. Give me 1 second. Let me make sure it's up here. I'm gonna put it in here too. And then I'll put it, I don't know, that's the only thing I can really find that kind of explained it. Did you guys see what I put in the chat box?
Mm-mm.
Oh, I see it.
Yeah. So, I mean, you can take a look at that. Hopefully it helps a little bit, and hopefully she posts some of the answers so we have a better idea. But,
Maybe, yeah.
other than that, I don't really know what else to say.
I'm not either.
Are you guys good?
I think so, except for that one, but yeah, we'll figure it out.
I'll post the link on iLearn too. Did you guys see the group chat thing?
Mm-mm.
It like disappeared.
Oh,
Wait, what disappeared?
my
The
link.
little YouTube link.
Oh, I still have it up.
I'll
I
put
don't
it.
know.
I'll put it on the discussion board too.
Okay.
All right.
Yeah.
I'll post.
Thanks for all the help.
Yeah.
Thanks, guys, girls.
Have a good night.
Mm.
Happy finals week.
Oh, thanks.
Happy finals.
Bye.
It's the last time we see each other.
Oh, it is sad.
Aw.
Oh, that's okay. We can still. I don't know. If we really need a genetics reunion, we can just get together on campus or something and just have a genetics learning party. Yeah.
I'll be in Hawaii. Sorry.
Wow.
Whoa!
Yeah, I'm gonna. So I guess I shouldn't put this. I wish I could. Let me stop this broadcast.